For most of history, when you asked someone, "How old are you?" the answer was pretty simple. You just count the years since they were born. But what if your body is aging faster or maybe even slower than the calendar suggests? Well, today, the fields of artificial intelligence and biology are starting to merge, giving us a much deeper and more nuanced way to understand how we age, moving way beyond the calendar to the level of our very cells.
And that brings us to the central question in a lot of aging research today. You see, the number of birthdays you've had, that's your chronological age. But the functional state of your body, its actual physiological condition, that's what we call your biological age. The thing is, those two numbers aren't always the same, and the difference between them can hold some important clues about our health and longevity.
So in this explainer, we're going to break down the science of predicting biological age. We'll start by defining these two different ways of measuring a lifetime. Then, we'll get into the machine learning models, or clocks, that use molecular and cellular data. After that, we'll explore some of the less invasive methods, and we'll wrap up by looking at the current challenges and what the future might hold for this kind of research.
Okay, first things first. Let's establish the two fundamental concepts that this entire field is built on: chronological age and biological age.
Chronological age is simply a count of time. It's also considered the single largest risk factor for major human diseases like cancer and neurodegeneration. Biological age, on the other hand, is an estimate of your body's condition. It tries to quantify the aging process itself by measuring very specific, age-dependent biomarkers.
So, how do you actually measure biological age? Well, scientists look for key molecular and cellular changes that are seen as hallmarks of aging. These include things like epigenetic alterations, which are changes to how our genes are expressed. There's also telomere attrition, that's the shortening of the protective caps on our chromosomes, and cellular senescence, which is when old, non-dividing cells start to build up.
And the main tool for doing this measurement is what's called an aging clock. Now, this isn't a physical device, it's a machine learning model. It's trained on huge data sets of biomarker measurements from people of known chronological ages. The model learns all the complex patterns associated with getting older and can then predict a biological age from a new set of biomarkers. The output from one of these aging clocks becomes powerful because you can compare it to a person's real age. This difference, which is often called age diff, gives you an indicator of that person's rate of aging. So a positive age diff would suggest accelerated aging, while a negative one suggests a slower pace compared to their peers. Now that we have a grasp of the concept of an aging clock, let's look at how they actually work. We'll start with the first major category, which operates by analyzing molecular data from what are called bulk tissue samples. Think of something like a simple blood draw.
Among the most foundational of these tools are the epigenetic clocks, specifically the ones based on DNA methylation. You see, methylation is a chemical modification to our DNA that helps regulate which genes are turned on or off. These patterns change in a predictable way as we get older, which makes them a very reliable biomarker for building accurate aging clocks.
If you look at the development of these clocks, you can see a clear technological progression. The pioneering models back in 2013 used simpler statistical methods to find these patterns. But more recently, the field has really shifted toward deep learning. Models like MethylNet, DeepMage, and Ultimage use complex deep neural networks, and they can uncover much more subtle relationships in the data, leading to significant improvements in accuracy. This number here, 2.15 years, represents the accuracy of that ultimate age clock. It's measured by something called mean absolute error, or MAA. Basically, that's the average difference between the age the clock predicts and a person's actual age. A lower number is better, and 2.15 years is a notable improvement over the earlier models.
The technology that's been driving these accuracy gains is the deep neural network, or DNN. A DNN is a machine learning architecture with multiple layers of computational nodes. Each layer transforms the data that comes into it, which allows the network as a whole to learn progressively more complex features and relationships. That capacity is what makes them so effective for analyzing complex biological data.
And it's not just about DNA methylation. These same deep learning principles are being applied to other kinds of molecular data. For instance, transcriptomic clocks analyze gene expression to see which genes are active. Proteomic clocks measure protein levels in the blood, which can be an indicator of inflammation. There are even microbiome clocks that use the composition of our gut bacteria to predict age. Each of these gives us a different window into the aging process.
All right, so far we've been talking about analyzing tissue averages, but what happens when we zoom in and start looking at individual cells? This is where things get even more detailed. This distinction between bulk and single-cell data is pretty important. Bulk data from a blood or tissue sample gives you an average measurement across millions of cells. The problem is, this can hide important differences between those individual cells. Single-cell analysis, on the other hand, lets researchers measure biomarkers in each cell one by one, which reveals a much more fine-grained picture of how a tissue is aging. This lets us ask a question that's very difficult to answer otherwise. Do all the cells within a single tissue actually age at the same rate? As it turns out, the answer is no. This variation is known as the heterogeneity of aging, and it's a key area of current research.
Looking at these individual cells has already given us some interesting findings. We now know that different cell types, even within the same tissue, can age at different speeds. This allows researchers to test anti-aging interventions and see exactly which cells are being affected. It's even shown us that people who live past 110, known as supercentenarians, seem to have delayed age-related changes in the composition of their cells.
Okay, up to this point, we focused on clocks that require a tissue sample. But what if we could measure biological age without having to draw blood at all? That's where non-invasive clocks come in, and they're particularly useful for large-scale human studies.
One of the most accessible examples of this is predicting biological age from an image of a person's face. By using 3D facial scans, machine learning models can be trained to estimate chronological age and even perceived age with a high degree of accuracy. This is possible thanks to a specific type of deep neural network called a convolutional neural network, or CNN. Basically, a CNN is trained on a massive dataset of images, say faces or retinal scans, that are labeled with a known outcome, like age. The network then learns to automatically identify relevant patterns and features, like skin texture or anatomical structures, and uses those features to make its prediction.
This table gives you an idea of the accuracy for a few of these non-invasive methods. You can see that 3D facial images can predict age within about 2.8 years. It's also noteworthy that a 3D scan of the brain can predict age with an accuracy of just 2.23 years, which is even better than some of the molecular clocks we talked about earlier. The clinical relevance of these measurements is significant. For example, research on retinal aging clocks has found that for every 1-year increase in what they call the retinal age gap, that's the difference between the retina's predicted age and a person's chronological age, there was an associated 2% increase in the risk of death from any cause. The practical advantages here are quite clear. Because this kind of data is easy to collect, these clocks are well suited for large-scale studies with thousands of participants. They hold potential for use in routine medical checkups to detect health risks early, and they can help researchers uncover links between biological age, lifestyle, and disease.
So this technology is clearly powerful, but it's not without its limitations. For our final section, let's take a look at some of the current challenges and where this research is ultimately headed.
One of the bigger challenges right now is integrating multimodal data. You see, aging is a complex process that affects the body on many different levels. A single data type, whether that's methylation or facial images or clinical tests, only provides a partial picture. The goal is to combine all these different data types to create a more holistic and accurate composite aging clock.
Another major hurdle is model interpretability. Deep learning models can be highly accurate, but their decision-making processes are often opaque. They're often referred to as a black box. For clinical applications, where you really need to understand the why behind a prediction to trust it and act on it, this lack of transparency is a pretty significant barrier.
To get around this, researchers are developing methods to explain what the models are doing. One such technique is called SHAP. It's an approach based on game theory that calculates the contribution of each input feature, like a specific DNA methylation site, to the model's final output. This helps to identify the most important biomarkers that are driving the age prediction.
And all of this work brings us to the ultimate purpose of this research. Being able to measure biological age is a notable scientific achievement, but it's really just the first step. The real goal is to use this knowledge to develop effective interventions that can improve health and maybe even slow down the aging process.
In the future, it's expected that these aging clocks will provide direct clinical guidance. They might be used to predict an individual's risk for specific age-related diseases, to help guide personalized medical and lifestyle choices, and to evaluate how well treatments are working to improve a person's health status. The ability to precisely quantify the aging process opens up some profound possibilities. It could shift the focus of medicine from treating diseases that have already taken hold to proactively managing the aging process itself. The long-term medical and societal implications of this technological shift are extensive, and we're really only just beginning to explore them.